Yeah, while we are still waiting for people to tune in, and I just want to start asking about your career trajectory. And yeah, we're very happy to invite Blair Benham-Pyle to share her work and her career with us today. So, Blair, actually, we overlap a little bit. It's a little bit surprise. I look up your experience. You were undergrad in Yale, and then the final year of your college, you went to Shanghai, and you did a research for about a year. Is that a full year?
Yeah,
And
yeah.
you were in my college. Actually, you're in my institute. You probably share a lab space. So you're just sitting. Maybe you are downstairs. I don't know. Like we are in the same institute, in the same lab, actually. So it's really surprised how the destiny brought us together, and how we overlap, and other people. Like it's you went on to Stanford to study cancer biology. Our institute was in development biology. So it's kind of exciting that you're coming back then for postdoc you to study more like developmental biology in the model system of planarian, and yeah, did a uh, great work in each phase of your career, and now you are studying like since 2022. I heard you studying your lab, your own independent group in Baylor. So just maybe before you even start talking about this paper, and just to share a bit, maybe it would be great to share with us a little bit how you think about the like what's the thinking now before you were doing the cancer biology, and then like in the beginning was mammalian, right? In the human or mouse model, like cancer tissue culture mostly. But then you start thinking about moving on to the very different, unique system of planarian. And how did you weigh your choices? And I mean, it's a bit of take a bit of courage to do so. Like nowadays, like it seems that especially the overall emphasis, like you see in biomedical science, we emphasize on translational medicine. And was there ever any concern about like the human relevance, or like concern about even just the resources, because fewer people are working on the planarians, right?
Yeah. Well, first off, thank you for the invitation. It's always really fun to get to talk to students and trainees about sort of basic discoveries. Um, but yeah, to answer your question, I did my PhD in cancer biology. I was doing mammalian cell culture and like epithelial cell lines. And I was actually coming from a biochemistry background, so I was interested in why, like essentially, how do cells interpret physical forces and how does that contribute to their behavior in tissues? And that question, led me to some really interesting biology, cell-cell adhesion complexes, like real cell biology kind of research. And, what I started getting interested in is not how sort of individual clonal cell lines behave in a dish, but how the collective properties of a bunch of individual and heterogeneous cells led to tissue morphogenesis. Because, we were seeing basically collections of cells in a dish respond really differently to very subtle changes in mechanical forces and sort of tissue densities. And I wanted to see how that played out in a larger tissue. And I was particularly interested in adult tissue morphogenesis because as a cancer biologist, you always want to figure out how to stop a cancer once it's formed, but truly, if we want to improve human health, it would be great to understand how we can sort of cut off cancer before it ever gets started. And so for me, I just got fascinated by the concept of regeneration. So in adult tissues that can regenerate, there are these sort of transient waves of wound-induced signals that get turned on for short periods of time and allow this remarkable morphogenesis. So replacement of missing or damaged tissues over a short period of time. And commonly, the signaling pathways that get turned on for a short period of time during these regeneration events have a lot of overlap with the same strategies used by cancer cells to take over a tissue and sort of migrate and grow. And so I wanted to understand how could organisms achieve regeneration without increasing the risk of cancer. And so I just started looking around for really sort of outliers in the regeneration space. And honestly, once I found planarians, they were completely counterintuitive to me as a cancer biologist. I did not understand how an organism could achieve a life cycle strategy where it really only regenerates. Like it doesn't sexually reproduce, it just rips itself into pieces and regenerates. And so I'll talk about that a little bit in the beginning of my seminar. But essentially, once I found planarians, it just did not make sense to me that this sort of bag of cells contained by body wall musculature could self-organize and regenerate after almost any injury repeatedly again and again without ever giving rise to cancer or stem cell senescence or aging. So, yeah.
And you have been trained, like before that, you were in Yale and the East Coast, and then you went to Stanford, West Coast. And then like you decide to go to Stowers Institute, which is in Kansas City, which might be unfamiliar to many people, like especially working on science. And what do you think about the environment? Do you still feel connected there? And how like do you do it takes your visit to convince you that you can do great things there, or you already know what they are up to, like what they can do over there?
Yeah. Yeah. So, I mean, fundamentally, when you're choosing your postdoc, you're really choosing your PI. And so, when I found planarians as a model organism, there were really only a couple people in the country that I considered. There were three HHMI investigators that all studied planarians: Phil Newmark, Peter Radin, and Alejandro Sanchez Alvarado. And, Alejandro was kind of like the start of it all. Um, and so and I was honestly very intrigued by the idea of going to Kansas City because, the cost of living for a postdoc starting in 2016, before like the salaries went up, it's tough to live in Boston or sort of coastal cities. And Kansas City is really not very expensive to live. And so, I went to the Stowers Institute, and the reality is like the resources at that institute are incredible. I mean, a ton of cores, lots of hard money that support the labs. And, it really is an institute designed to help you do amazing science quickly. And they're not necessarily constrained by some of the ideas of, you have to have NIH funding, it has to have a translational impact. You really are encouraged to pursue curiosity-driven science. And so once I visited Alejandro's lab and saw all the core facilities there and got to explore Kansas City a little bit, I just felt like it was a really good option for a postdoc. Alejandro has a track record of helping people find independent research laboratory positions. The Stowers Institute is really well equipped to do science. Kansas City is a really easy city to live in. There's like good entertainment, good food, good music. Like housing isn't expensive. You could buy a house if you wanted to as a postdoc. Um, and so that was perfect for me. And Alejandro is the kind of PI that just really lets you pursue your ideas and encourages you to kind of think big. Like, what is the right experiment to answer your question? And if you see something interesting, go for it. So that's what I was looking for. Oh, sorry, you're muted.
Oh, sorry. I just mentioned that you appreciate this freedom to pursue like curiosity-driven research. And one other thing, I mean, I wonder if it's ever any concern, like because it's a smaller community and maybe there are more limited resources. Do you have to like coordinate, or do you need to see if you want to do something, like do you want to look around, see like because community maybe you all know who is who, and so that you do not end up in redundant efforts, or like is that not really a concern? What do you see this?
Yeah, it's not really a concern, to be honest. There's 20 some investigators at the Stowers Institute, and they all study dramatically different things. Um, and so, half of the staff are in labs themselves, and then nearly half of the staff are in what's called research support roles, which means they run the cores. And they are professional scientists that just help the labs do the most cutting edge research they can. And so when I saw a new technique that came through, like split pool ligation based sequencing or spatial transcriptomics, like SlideSeq, I would just go to the cores and say, okay, histology core, can you help me section onto this slide that has beads with barcodes? Next gen sequencing core, can you help me like adapt these library prep protocols to planarian tissues? And, they were fantastic and they would help me optimize the protocols. A bunch of the core members are on my papers out of the Stowers Institute. And so, I mean, yes, it's a small community, but it's connected to so many different networks, sort of nationwide. And all of the PIs are very in tune with what their fields are doing that I never really felt restricted. And you can go to meetings to meet more people, right? And
Yeah.
so most of your postdoc is spent at the bench. So as long as you can do your experiments that you want to do and you have teams of people to help you do those experiments as quickly as possible, you're really set up to succeed, in my opinion.
In terms of the model system, like of a planarian, is there anything, any technique, any cool thing that is not yet possible that you wish to be possible in planarian? Because I know Charlotte is a student, maybe she's also considering postdocs in sometime in the future. Like, and
Yeah.
other people who see the recording later, like any.
Yeah, absolutely, absolutely. So there's two critical technological gaps in well, three. There's three technological gaps in planarians right now that really sort of separate us from more traditional model organisms. That's gene editing, so the ability to edit the genome, live imaging, which is you kind of need genetically encoded markers for live imaging. Sometimes you can get away with dyes, but the worm is pigmented, so it'd be nice to be able to fix some aspects of that. And then the last one is actually ex vivo stem cell culture. And I think those three methods are probably going to get combated in the next decade or so. So the worm itself is biologically challenging for some of them. So the stem cells are tightly regulated by their environments and by sort of the surrounding tissues. And so getting them to behave outside of the context of the worm is really hard. But the more we understand about their microenvironments and regulation, the more we can maybe get them to expand ex vivo, which would allow us to do lots of cool things like clonally expand individual colonies of stem cells, do sequencing, track mutations, like all sorts of cool stuff. Live imaging, I would love to watch regeneration in planarians live. We've now figured out ways to depigment the worms, which is exciting. We can maybe get dyes, like some really good microscopes. I feel like this is coming very soon. Um, and then with transgenesis, so the real issue with planarians is that we don't have an embryo. So as I'll talk about in my seminar, planarians are asexual, obligate, at least the strain we study is an obligate asexual. And so we don't have an embryo. We have little regenerating clonal progeny and a ton of stem cells that build up the tissues in those animals. So the best strategy to make a transgenic worm is to infect or inject gene editing materials into stem cells and then select for the edited stem cells and get them to expand, either in vivo or in a dish, and then transplant them back into an irradiated worm. The problem is that, in an organism that relies on a clonal expanding stem cell population, they actually have a ton of defense mechanisms to prevent genetic editing and mutation in the stem cell compartment. And so we basically have to figure out strategies to evade their innate immune system and cell competition to ensure that our edited stem cells are the ones that actually take hold and make a clonal line of planarians. And that is actually not an easy, not a trivial problem to confront. So
Because when Alejandro came to Rockefeller, I asked him if there were endogenous viruses on for planaria that you could leverage, and he said that that wasn't the case. But I was
I
very
don't think
shocked that there
I think
aren't
there
any, and
are.
I
So
wonder
there
what
there
your thoughts
are
were on that.
yeah, there are endogenous transposons.
Okay.
So you could go after some endogenous transposons, and they hop around in the genome even actively.
Uh-huh.
And so you could go after an endogenous transposon system. There are also there are viruses that infect planarian strains. There's actually a preprint from Bo Wang's lab where they did a chimera between asexual and sexual worms, and they used a virus as a way to detect the strains. Um, and so you can go after viral components. I'm not sure why
Hmm.
no one has talked about trying AAV delivery in the planarian community. Maybe you need like maybe it's just no one wanted to figure out the BSL-2 like status.
Uh-huh.
Or it could just be that someone tried it and it was a negative results. Um, there are also labs trying planarian liposomes to
Uh-huh.
deliver genetic material. So, I mean, I really think that there are lots of labs working in parallel trying to figure out this
To
problem.
figure out this problem. Yeah, it makes
Yeah.
sense.
And so I think, for a young PI like me, I'm mostly in the figure out the experiments I would want to do and figure out which component I'm most capable of contributing to. And so because of
Mm-hmm.
my cell culture background and my interest in sort of stem cell niche regulation and that kind of stuff, I've sort of tried to, I think the most likely thing I'm going to go after is live imaging and clonal expansion of stem cells ex vivo.
Mm-hmm.
Because I also just don't
Yeah.
trust a transgenic stem cell to expand the way I want it to in vivo, given everything I now know about intercellular signaling in the worm.
Even the live imaging. I mean, you can live image a mouse, and that's certainly not, highly pigmented. And so I feel like, sure, you might not live image the whole, right? But you'll have some depth, but you can still do some live imaging, right? I guess the fluorescent reporters are the harder component
Yeah,
to
so
that.
it's the
Mhm.
reporters that are the issue.
Yeah,
So you need to
yeah.
figure out like
Some way
dyes
to get some dyes
or
that
yeah,
come
or
in.
transient expression. So
Yeah.
like transient delivery of fluorescent dyes. Um, I actually think that's quite possible. And we can actually test some very cool hypotheses just by watching cells migrate and divide and fill into different tissues during regeneration. And
Yeah.
so that's,
Okay,
I think we
Blair,
can like to start maybe.
oh yeah.
I don't know, Charles, what your thoughts are on this.
sorry, I was just about to say when we finish this question, I'm going to let Blair start her seminar and we can then have a Q&A. But yeah, please, what did you trying to say? What were you saying?
No,
And
I was saying that like I think we can let Blair start her seminar, no? Like I think
yeah,
we can.
yeah. Then we can continue our
Yeah.
conversation and
Hmm.
we might have some question about the seminar.
Okay. Thank you. Thank you.
Thank you.
Well, let's play this. Okay, does everyone see the slide with like the title slide? Okay, perfect. So what I'm going to talk about today is essentially the story that Charles asked me to talk about, which is Chris Arnold and I's discovery of sort of how planarians asexually reproduce, and specifically Ashmatea mediterranea sexually reproduces. So the first question, so bit of background on me. I'm going to go through this pretty fast because I kind of talked about it already. So I'm a cancer biologist by training. One of the things that's always fascinated me is how diverse cells rebuild and maintain an organism and how that can go wrong. So I often think about this in the context of maintenance or tissue turnover, or even in the context of tissue aging. And then in the more extreme circumstance, we can think about how organisms rebuild missing tissues in the context of regeneration. Now, this is basically all of these processes are regulated by the balance between cell division and cell death, growth and removal of dying cells. So addition of new cells would be cell division, differentiation, sort of stem cell self-renewal processes, and how tissues are maintained across the lifespan of an organism. When things work really well, you can get things like tissue regeneration. But of course, when cell growth is misregulated, you can get something like cancer. Now, on the other side, with cell clearance or cell cycle arrest, You can experience cell death processes, stem cell senescence, decreased growth within a tissue. And under good circumstances, mechanisms that regulate cell clearance and arrest can actually play an important role in tumor suppression. But on the negative side, it can lead to degenerative diseases or aging. So, when I started my postdoc, I was looking for cool organisms that would allow me to understand sort of extreme resilience mechanisms and extreme adaptations for improved growth degrowth balance. That led me to planarians. As most of you may know, planarians are very well known for their regenerative abilities. So you can basically cut them up into 3, 4, 10 pieces. And these individual pieces of tissue will wound heal over the course of maybe 4 to 6 hours. And then they'll slowly build blastemas in which stem cells can sort of differentiate into all of the missing components of the animal, eventually building out the sort of unpigmented blastema and rebuilding any missing tissues that may have been lost in the course of the injury. And this process takes anywhere from 7 to 14 days. By the time you get to sort of 12, 14 days, the animal has actually kind of completely remodeled, reshaped into a smaller version of the original animal that it was taken from. So, this process is driven by an abundant stem cell population where you cut off this sort of tissue fragment, the stem cells migrate and proliferate near the wound site and eventually rebuild all missing tissues. Now, of course, planarians are not just stem cells. They actually have diverse tissues that are very homologous with mammals. So, you have an abundant stem cell population, but you also have a branching, polarized intestinal system. You have sort of diverse muscle cell types across the animal. There's actually small protonephridia-like structures that highly resemble a kidney, or similar to a Drosophila kidney, really. There's a beautiful central nervous system that's also associated with the peripheral nervous system. And then there's all sorts of different sort of phagocytic and secretory cell types that fill the mesenchyme of the animal. And so, what's beautiful about these tissues is that it's not just the stem cells that respond to amputation, it's these differentiated cell types as well. So when you cut an animal, the pre-existing tissues actually remodel and reshape into the smaller body plan while being sort of integrated with newly regenerated tissues. And so, in the case of the intestine, for example, a lot of the morphogenesis that happens with these intestinal branches coming together is almost independent of stem cell proliferation. And so it's really unknown how these post-mitotic tissues and pre-existing tissues are reshaped and remodeled within the context of a regenerating body plan, which is an interesting question. So why would a flatworm ever choose to be this regenerative? I mean, obviously regeneration is a good trait, but this is pretty extreme for an organism. And so I think it's always nice to mention sort of the diversity and evolution of why this might have happened. So planarians are actually a huge family of flatworms that can be found all over the world in all sorts of marine to freshwater environments. And they actually are not all great regenerators. So some planarian flatworms can't regenerate at all. But Schmidtea mediterranea is actually one of the best regenerators in the entire family. You can cut them anywhere along their body plan and they can rebuild any type of tissue. So the closest relative of the asexual strain that we study is actually a sexual strain of Schmidtea mediterranea that was co-isolated with the asexual strain in Barcelona, Spain. And the sexual strain pretty much looks exactly like the asexual strain. It grows bigger and it develops germline. So it actually lays embryos into these little cocoons. And what we found is that at some point in evolutionary history, there was a chromosomal translocation that moved a chunk of chromosome 1 over to chromosome 3. And this essentially broke germline specification. So in Schmidtea mediterranea, chromosome 1 contains a bunch of the genes that are important for germline. And because a large chunk of them got moved outside of their chromosomal context, the animal can no longer create mature ovaries and testes, and it can't make an embryo anymore. And we don't know how long ago this happened, but we think tens of thousands of years. So this worm suddenly was faced with a choice. Either it learned how to live forever very, very quickly, or it found a different way to produce progeny. And so its strategy was to sort of upregulate this behavior called asexual reproduction, where it rips off a piece of its own tail, and that tail regenerates and repeats this process over and over and over again. And so what was fascinating to me as a cancer biologist is that essentially you have this worm trapped in an endless cycle of growth and regeneration, fueled by an abundant pluripotent stem cell compartment. Yet, as far as we can tell, we've been expanding these clonal worms for 2 decades in labs all over North America and Europe. As far as we can tell, we have never seen a decline in regenerative capacity, and we have never seen tumors develop in our lines of worms, which did not make sense to me. So now my lab is interested in studying essentially how diverse cell types across the organism are coordinated to achieve regeneration, and how does regeneration intersect with cancer and aging resistance. But of course, the reason I'm here to talk to all of you is to talk about this paper that Chris and I wrote, which was the first story to come out of my postdoc and the second and third story to come out of Chris's postdoc. And this is essentially, why do planarians fission? So this is a behavior that seems to have been upregulated specifically in planarians that don't have the ability to make germline, or who are very, very bad at making germline. So very infertile animals. So why would you upregulate a behavior like asexual reproduction in an animal that either cannot make embryos or is very bad at it? And so, how does an animal know when it wants to asexually reproduce? And, as a cancer biologist, I was really interested to understand if this was some sort of size control mechanism in planarians. So when I joined Alejandro's lab, I was like, I really want to study fission. It seems odd, it's weird, but I want to know why an organism would develop this behavior and why it would emerge in the species. And now this is where Chris comes in. So Chris had been studying basically the microbiome of planarians and how the innate immune system detects infection and how the microbiome responds to regeneration. And as a part of his experiments, he had been doing some treatments with antibiotics. And one of the phenotypes he noticed with antibiotics was that he thought he was seeing different rates of asexual reproduction. And so he had started thinking about designing assays for fission behavior. And I was like, that's perfect. I also am interested in fission behavior. If we team up for the assays and try and do the initial characterization together, then we can kind of go off and pursue our separate questions as postdocs in our own labs. And so, basically we decided to team up. And so the first question we really wanted to ask is, what is fission behavior in planarians? Can we induce it? And this is actually where sort of serendipity in the Stowers Institute kind of works some magic. So the Stowers Institute had needed to build up massive quantities of planarian tissue. Alejandro had a big lab. And so they started designing, previously everyone raised planarians in Tupperware containers. And so the Stowers Institute decided that they didn't want to do the manual labor of raising planarians in Tupperwares. They wanted to build aquariums to house planarians in large quantities. And so they built these sort of recirculating aquatic systems that can house hundreds of thousands of planarians. And they started feeding them chunks of beef liver that they would just drop into the aquarium. And this allowed us for the first time to raise planarians with a very high feed rate and basically constantly recirculating water, so we could grow them to a much higher density without the population getting dirty. And because of that, they got really big, bigger than most planarian labs ever study. So most planarian images you might see in a paper before this one, the worms are maybe 2 to 5 millimeters long. But we started growing planarians to be 15 to 20 millimeters long. And it turns out that when you have a really big planarian that suddenly comes out of an environment where it's very well fed, and you put it in a dish, all it wants to do is fission. And so we found that we could take these large worms out of the research system and put them into live imaging environments, and we could catch live for the first time this fission behavior. And so we saw that the worms would rip off a small piece of their tail. It was an incredibly asymmetric process. And this process took maybe 24 minutes. And when we took animals out of the research system, they would do this behavior at least once a day, often multiple times per day. So this sort of led to the first question, which is, if no one has been observing this behavior previously, maybe it's because it's size dependent. And so we first asked the question, let's take small worms and big worms and do the same assay, take them out of the research system and see what they do. And what we found is that over the course of 2 weeks, a big worm produced many, many progeny, and a small worm might produce 1 or 2. You could quantitate this. It's a beautiful linear relationship. So basically, once they hit 5 millimeters or in size, you sort of get this linear increase in the number of progeny they produce. And this is actually because the time between asexual reproduction events gets tighter and tighter and tighter, the bigger an organism is. One of the other interesting observations we made is that the progeny size is always the same. So it is independent of how big the worm is. They always make a fragment that is about 1.2 millimeters long, which is also really interesting, but makes sense when you realize that these worms have to fission multiple times in 1 or 2 days. They don't actually have time to regenerate between these asexual reproduction events. And so an individual worm will actually produce multiple sort of set pieces of tissue that are all the same size from its tail all the way to its head over the course of a couple weeks. Now, we were pretty shocked at how regular these sort of fragments of tissue were. And it got us really interested in how they might ensure that fission fragments are always the same size. And then, so basically, we had been working on this project for, I don't know, 4 months, 5 months together. When Chris suddenly came to me in lab, he's like, okay, this weird thing happened. I was like trying to immobilize a worm underneath a cover slip, and I accidentally squished it. And I think I'm seeing something. Could you just, I feel like when I squish the worm, I'm seeing lines, but that doesn't make sense. So like, can you do it and see if you also see this? And I squished a worm under a cover slip and it looked like there might be lines. So I got out my iPhone and videotaped Chris squishing a worm underneath a cover slip. And that iPhone video turned out to be in our Nature paper. And it's because when you squish a worm underneath the cover slip, you can actually reveal these mechanically vulnerable planes in the body wall musculature and epidermis of the animal that almost perfectly fit with where we would expect fission fragments to be sort of fragmented in the animal. So Chris went on to do all sorts of compression assays where he could squish worms of different sizes. And indeed, we saw that the bigger a worm was, the more these compression planes got patterned along the body axis, and they perfectly aligned with where the animal naturally produces a fission fragment. So this was pretty exciting. So because they were patterned along the anterior posterior axis, and they kind of looked like segments, the first thing we wanted to know is, could it be that anterior posterior patterning controls where these fission planes are deposited? And so in planarian literature, there's a bunch of signaling pathways, developmental signaling pathways known to regulate anterior posterior patterning. And so we decided to knock them all down and see if they affected fission behavior. So the way we did this is we would take animals out of the research system. We would put them in these little flow-through RNAi feeding cups, feed them RNAi food. Then we would take them out of the research system, put them in a dish by themselves, measure their length, count their fission fragments every day for the next 2 weeks, normalize and sort of colorize the number of fragments per body length that they made over those 2 weeks, and produce a little time course for each worm of how it produced fission fragments. Then we would do this for multiple worms and create sort of a little heat map of fission activity within the RNAi treatment. So something that's really blue does not produce a lot of fission fragments over 2 weeks. Something that's really yellow does. So the results of the assay gave us a huge range of fission activity when we disrupted anterior-posterior patterning. And we found several things that were significant. And what we saw was that when we knocked down sort of activin and Wnt signaling molecules, we saw less fission activity. And when we knocked down inhibitors of Wnt like APC, we saw an increase in fission activity. And so you can sort of see this when you look at the parents after several weeks. So the worm on the left is the worm at the beginning of the experiment. The worm on the right is the worm at the end. And you can see that some worms have gone through most of their body tissue, but some worms really haven't fissioned any of their body tissue off at all. So the next question is, okay, great. Do they regulate fission planes? And unfortunately, the answer is mostly no. So we knocked down all sorts of Wnt components, saw really, really strong fission phenotypes, but didn't see any change in where these compression planes were patterned. We saw a subtle difference when we knocked down TGF-beta components, but the fission phenotype we saw is significantly stronger than maybe a 50% reduction in the number of fission planes would account for. And so we didn't actually think that Wnt and TGF-beta were initially working through these compression planes. So that sort of led us back to the behavior itself. So behaviors in planarians are controlled by the central nervous system, and then these sort of two anterior cephalic ganglia that connect to ventral nerve cords in the animal. And when we looked at where the genes in our screen were expressed, yes, they were expressed sort of in a gradient along the body, but if you looked at the brain, they were actually very highly expressed in the brain tissue itself. And so that led us to this question, like, could it be that these polarity genes are not just controlling sort of axial polarity across the whole animal, but are actually coordinating behaviors through the brain? So to test that, we actually had to come up with a behavioral assay. So we started videotaping planarians in a sort of 6-well grid system. And if a worm successfully fissioned, we would create this uptick on a timeline. And if it attempted to fission but didn't produce a fission fragment, we would do a downtick, sort of an unsuccessful fission. So what this looks like for a control animal is if we watch them over 9 days, they would sort of have a couple successful fission attempts, one unsuccessful attempt, a successful attempt, an unsuccessful attempt. And so the frequency of these attempts and the sort of success ratio could tell us something about the fission behavior over time for each individual worm. And what we found is when we knocked down Wnt and TGF-beta components, we could actually decrease or increase how often the worm tried to asexually reproduce, which really means that these genes were affecting rates of behavior more than the ability of the worm itself to rip itself into 2 pieces, which was really surprising to us because we had expected polarity genes to be controlling these fission planes. So we quantitated this, it was highly significant. And that kind of led us to the question is, how could these polarity genes be regulating behavior? And, you know, the obvious hypothesis might be that patterning genes coordinate patterning of the brain, and patterning of the brain could influence behavior. So we did a sort of in situ screen of all sorts of different neuronal subtypes that are known to exist in planarians. And we actually got a couple gene markers for mechanosensory neurons that popped for us. And these are specific cell types that seem to be patterned in accordance with size. So PKDL2 is a homolog of PKD in mammals. So it's a mechanosensitive channel. And it marks sort of this population of neurons that stretch along the edges of the worm, along this like sort of ventral race stripe, as we call it, which helps coordinate swimming behaviors, and then a little bit at the front of the worm that sort of helps sense mechanical forces in the environment. And then the other type is this GABARG positive mechanosensory neuron, which is also an ion channel. And they sort of form this band of neurons around the front of the head. And we found that the patterning of these populations is actually size dependent in the worms. So then we decided to test if our patterning genes also influenced how these cell types were patterned. And we indeed found that when we disrupted either TGF-beta signaling or Wnt signaling, we could see altered patterns of these mechanosensory neurons in the brain. And then finally, we wanted to ask, do these mechanosensory neurons regulate behavior? So we knocked down the ion channels that marked the neuron populations to see if they altered vision behavior. And not surprisingly to us, we found that when we inhibited these mechanosensory ion channels, we actually saw increases in fission behavior. So we think that mechanical stimuli inhibit fission behavior because it suggests it might not be a safe environment for the planarians to asexually reproduce. And we also saw this in our behavioral assay. So when we knock down GABAerg, we see this incredible increase in the frequency of the fission behavior. And so we think that the mechanosensory neurons are patterned so that the larger worms are a little bit less sensitive to mechanical forces in their environment and slightly more likely to asexually reproduce. And when you mess up patterning of these neurons, it essentially makes the bigger worms more sensitive to mechanical stimuli, and they're less likely, or sort of more likely to asexually reproduce. So what we learned in this story is that planarian fission is a growth-dependent behavior, that fission planes are pre-patterned and pre-established along the anterior-posterior axis, and that Wnt and TGF-beta factors are important regulators for patterning the brain and coordinating this behavior with size. And so fundamentally, this paper launched sort of a lot of really interesting questions about how sort of environmental and intrinsic factors are integrated in a worm to generate this growth-dependent behavior. And Chris actually went on to study the Hox genes and found that Hox genes are important for patterning these fission planes along the anterior-posterior axis. And he's also following up on various types of transcription factors that couple regenerative capacity to asexual reproduction and behavior. How am I doing on time? Okay. I'm going to do a quick vignette of like forward thinking stuff. So my lab is in a medical center, and in a medical center, we kind of have to justify why we want to study a weird worm behavior. So I'm going to tell you briefly, sort of returning to some of my central questions, why would an organism evolve asexual reproduction behavior in the context of not being able to make offspring anymore? And this sort of feeds into my initial question of like, how does a worm know when it's time to asexually reproduce? And is this a size control or disease resistant mechanism? So one of the first things we did is to really ask fundamental question: under what circumstances would an animal want to asexually reproduce? And what additional signaling networks beyond patterning of the brain might regulate this behavior? And so to go after this, we've done sequencing now on small versus large asexual and sexual worms and looked for genes that are specifically regulated with size only in the asexual animals. And we found that genes that are upregulated with size only in asexual animals are associated with neural stress and inflammation, including like amyloidosis and sort of protein aggregation. We found that the things that are downregulated, quite surprisingly, almost all have to do with muscle growth and telomere maintenance, which really suggests that some of the phenotypes that we're seeing changing with size, specifically in asexual animals, are associated with pathways commonly correlated with disease or age. And so that's led us to sort of a new hypothesis, which is that asexual reproduction and subsequent regeneration is actually an adaptive behavior to help the animal reset whenever it becomes too stressed or too damaged to continue growing. And it's actually a really interesting adaptation for an animal that doesn't have the ability to produce offspring, because you need to sort of reset your lifespan if you've encountered a stress or damaging environmental factor. So the new hypothesis is actually that one of the components that feeds into this behavior is actually stress and aging. And so we're beginning to starting to test this hypothesis. So are there any hallmarks of age that decrease or decline with, basically increase with age and decline after a generation? We've looked at small versus large animals and the regenerates taken from aged large worms. We've looked at stem cell quiescence. So in a small animal, a large fraction of the stem cells are actively proliferating. But in a large worm that's very likely to asexually reproduce, the percentage of stem cells that are proliferating declines. But in the regenerates from that large animal, you see this stem cell proliferation increase again. And this is highly significant. We've also started using quantitative telomere fish to look at telomere lengths in the animals. And we find that as they get larger, telomere lengths become much more variable across the animal, but seem to be reset after regeneration. And so this really kind of starts to hint at this idea that asexual reproduction might actually be an age-dependent or damage-dependent phenomenon, which is exciting because it actually means we can use fission behavior and rates of fission behavior to identify genes and pathways that either mediate aging or damage control in the animal, or the detection of aged tissues by the planarian central nervous system. So we started a massive RNAi screen for fission behavior where we feed animals RNAi food, we isolate and starve them in a dish, and then we watch and quantitate how many fission fragments they produce over 7 days. Now, a normal worm will produce 5 to 7 fission fragments per animal over the course of 7 days. But if you knock down a gene really important for asexual reproduction, this number dramatically drops. We've now screened about 750 genes so far, and found a number of putative inhibitors and activators of asexual reproduction. But what's really interesting is that most of our significant hits are expressed by just 3 types of cells. There's intestinal and peripheral phagocytic cells that have a lot of phagocytic machinery and especially innate immune signaling machinery. There are neurons that control the behavior, and there are stem cells. And if we look at the genes expressed by these cells that also regulate asexual reproduction, we see stem cell replication and translation machinery. We see neural amyloids and amidated peptide signaling, and we see intestinal and phagocytic innate immune signaling. And so we now think that there is an axis where increasing damage and stress within the stem cell compartment is detected by the innate immune system in the worm, which then coordinates with the nervous system to trigger asexual reproduction and the opportunity to reset. So what's amazing about this is that we now think that asexual reproduction, this like weird behavior isolated to this highly regenerative worm, actually may be playing a really important role in the resilience of this organism to time and disease. We've found a few phenotypes that seem to correlate with age or stress, but we're now using our bulk sequencing data and RNAi screening to sort of really get at some fundamental mechanisms here. And we think we can now use asexual reproduction as a way to uncover novel mechanisms through which an organism can detect stem cell aging and then induce a behavior that could potentially fix that. And obviously, if asexual reproduction is an age-dependent behavior and an opportunity to reset the organism, that means that whole body regeneration is somehow rejuvenating the animal or resetting its biological clock, which would be a really cool thing to study, given that we now have 2 decades of knowledge of how regeneration works in this worm. And we can build on that within this new biological framework. So with that, I'm going to just acknowledge the Santos Lab at the Stowers Institute for launching this very, very cool project and allowing me to study asexual reproduction with Chris. And I'll plug that my lab is awesome at Baylor. So if anyone's interested in postdoctoral or graduate opportunities, we have lots of cool projects related to this, as well as cancer prevention in planarians and stem cell stress. So happy to take questions now.
That's really cool. I do have some questions, but maybe I will give others the opportunity if they want to. I want to say something. If not, I will start.
Feel free to start with your questions, Charles.
But maybe you want to say something? Sorry, Charlotte.
I'm thinking through questions.
Okay, you have some thoughts about it, just need to organize. Okay, so I just want to ask a couple of things. First, I think maybe related to the technical limitation, but when you show the neurons, you cannot see what the axons are. Is there a way to visualize the axons? Anybody that might work? I'm just curious whether the axon actually project towards the fission planes.
So there are antibodies that can label synapsin, so you can actually see synapses. There's some cross-labeling with some secretory cell types, actually, but they do, beautifully label sort of axonal projections in the animal, both in the central nervous system and connecting to the peripheral neurons. There are these like sort of midline crossing axons that are at regular increments in the worm, but the step size is like smaller than we would expect fission planes to be. However, I mean, I don't think it's unreasonable to think that since mechanosensory neurons regulate fission rates, it's possible other neuronal cell types might help pattern planes or fission planes along the axis, or that they might even be mechano-responsive. Um, it's an untested hypothesis, but you can visualize the axons, and then you could use in situ staining in combination with the antibody to try and figure out which types of neurons are most likely to interact with fission planes. Yeah.
Because so far you showed that other than the that mechanical vulnerability of the fission plane, we haven't really seen the molecular characterization of what is there, right? Like, is it possible to do a quick crush and then quickly micro dissect this to see how they was differentially expressed, what marks the plane?
Yeah.
And
So
also, there are
you can
several.
also fix an animal
Yeah.
while it's fissioning. So if you catch a worm in the middle of a fission event, like right before it's about to tear, you can fix the whole worm. And then you can actually see, I've done that once a couple times, and basically you can see that the body wall musculature is torn and a lot of the internal tissues are sort of like jagged. And the last tissue to break is the epidermis. And so you could label, try to figure out, basically dissect out just that piece and then do differential gene expression analysis, or honestly, maybe spatial transcriptomics would be the way to go at it, to try and find sort of this regularly intervaled gene or like molecular marker of the fission plane. Yeah.
Yeah, even though there are several fission planes, when you crush it all at once, they can be visualized. But when the sexual production happens, it's always the last piece that shut off first, right? So
Yeah.
it's only always the last piece, but not anything in the middle before that fell off, right?
Yeah.
It's curious.
We don't know why that is. When you watch them, we think maybe there's like an adhesive component in the most posterior section, and that like anchors the worm, and then everything else kind of like is pushed away from that last piece through a peristaltic like wave motion. And I don't know what it is precisely that defines this like last fragment versus others. It's possible Chris's lab might be working on this. He did find like an adhesive gland downstream of the Hox genes. And so it's possible that, the Hox genes are helping to sort of differentiate the segments from each other. And then when you rip off that last piece, they like shift up a segment.
When you are doing your screen, you don't really see weird phenotype like they just decide not to fission from the last end, but just break from the middle fission plane or other plane. Like other
So far, no. We did quantitate the size of the fragments overall. And so far, yeah, it really seems to be bimodal. We did get some fission phenotypes from the genes that was just like lethal. Um, or immotility. So, when you mess up neuronal structures, it's actually quite common that you mess up motility. And so you have to do a secondary screen where you confirm that the worm can still swim. And the reason it's not fissioning is not that it's just sitting there. But yeah.
Yes, I can jump in with a question. I think this idea of taking a stress and instead of like trying to survive it, just like restarting, whatever, generating more progeny is really interesting. Do you see like evolutionary disadvantages to this resetting rather than like giving the worm enough time to like adapt its behavior and then like transmit that behavior, whether it's through progeny or, you know, like learning or, yeah.
That is a really interesting question. So we have looked at sort of rates of asexual reproduction and with different doses of stress, mostly like ionizing radiation or something that we know affects the stem cells. And there's sort of this sweet spot actually where you see increased asexual reproduction behavior, and it's actually at sort of low levels of stress.
Mm.
If your worm is really stressed and really sick, it actually doesn't asexually reproduce. There's this like switch that happens where at that point, it actually focuses on controlling, especially, I think this is probably an adaptation to like an infection, where you don't necessarily want a worm infected with a virus that could affect the stem cell population, or maybe a really strong carcinogen in your environment has really compromised your stem cells and you don't want them to regenerate tissue. You essentially inhibit asexual reproduction and focus on repairing whatever the extreme damage is. And if you can repair the extreme damage and stabilize the system, you'll kind of like go back to growing and, you know, restart asexual reproduction. It's actually the really subtle stresses that seem to affect asexual reproduction rates. So it's the sort of really low level stress. It doesn't necessarily affect the number of stem cells or how they're proliferating, but it's enough for the innate immune system to be like, maybe we should not do this. Whatever it is we're doing, we're going to cut it off before it gets too bad. But in terms of like, why not learn? I think that's actually a really interesting question. It's unclear how well planarians learn, and how those memories might be translated or sort of passed to the next generation of planarians. There was one experiment done where they sort of associated a chemical cue with pain in the animal, and regenerated progeny still remembered the cue. And so it's possible that neurons that were trained in a prior environment that were sort of captured in the tail fragment that regenerates could sort of train the new brain to still sense that chemical cue. But whether you could get like sort of a complex behavioral adaptation, I don't know. But I think, yeah, I mean, the worm itself is like maybe not complex enough to have I mean, they might, I think they sense population density. It's hard to know if they like work together. I think they can trail each other to find food based on their behaviors in our aquariums. They like leave these mucus trails that I think other worms can kind of follow. But realistically, it's probably easier for them to just disperse and start again than to try and sort of rapidly develop a behavior that would allow them to adapt to their environment. Also, when you asexually reproduce, it's also a dispersal strategy because basically they drop a fission fragment and then swim for 4 hours and then drop another fission fragment and swim for 4 hours. So when you watch them in the wells, it's actually pretty obvious that they're actively moving between fission events. So if they're in a bad environmental position, going through subsequent asexual reproduction events is a way of dispersing your progeny into different environments and potentially escaping. So
That's fascinating. Thank you.
Can I ask you a very, like, I'm not sure I like really thought this out like very well, but like it's a very naive question. Because like my understanding of like these planarias that they were all coming out of this one like fountain thing in Barcelona and they were like fished out. And one of these planaria, like whatever was really interesting. And so that's kind of how it started the whole field. But so we're really studying like this one worm and all of its progeny. And I'm wondering, like in the wild, where there is maybe more like environmental cues, because like I mean, if you keep them in an aquarium, right, like then there's no real external challenges. But I'm wondering, like in the wild, like if you were to fish out all of these planaria, like how heterogeneous would they be at a population level? I'm just curious. I mean, I know it's not necessarily related to, I was just having some, yeah, I just was thinking
Yeah,
about
no,
this kind of stuff.
that's a great question. So it's true. Most of the planarians derived in North America are derived from like a handful of worms that Alejandro and Phil pulled out of a fountain in Barcelona. And they isolated both the sexual strain and the asexual strain and karyotyped it early on to find these two diploid genome types. The original discovery of Schmittea mediterranea in the 60s in Spain, they actually found three karyotypes. They found a diploid sexual, a diploid asexual, and a triploid asexual. And in theory, all of those wild populations could still exist today. Um, Apparently, Barcelona fixed its plumbing, so the fountain specifically, you can't really sample anymore. There aren't any planarians in it. But if you go out into wild streams around Barcelona, you can still find wild strains. And there are sort of similar species. So there's like this family, Jugacia and Gerardia and Schmittea, that are all highly regenerative. And they're all pretty similar morphologically. So Jugacia polycroa looks very similar to Schmittea mediterranea. You can tell them apart, but they look really similar. And there was a recent genome paper from Jochen Rink's lab that looked at sort of chromosomal evolution between these different species. And he did find evidence of like high repetitive DNA sequences and sort of elevated levels of chromosomal translocations and rearrangements. And so there might be a downside to regeneration and like this lifestyle strategy, which is that when you're kind of highly dependent on a really abundant stem cell population, it means that if your stem cells mutate, one, you need ways to like make that work for you, but also you are very likely to kind of have a sudden mutation occur that becomes fixed in a population. And so I think future work is probably going to really try and understand like how can you have increased genomic instability while also still maintaining sort of the robustness of the organism itself. And so, I'm sort of interested in studying like the innate immune system and how it detects damage, as well as like how that's integrated into sort of cancer prevention and regeneration and morpholaxis and morphogenesis. But other folks are really going to focus on, I'm sure, DNA damage mechanisms. Like, how is it that the genome itself is repaired and maintained? And are there mechanisms to ensure that, even if there is a translocation, you find the stem cell that has a stable translocation that you can work with and pick that one and get rid of everything else? And so I think that's sort of a really interesting question. I think Jochen Rink's lab has the most strains of planarians. He sort of made it his mission to collect as many different strains of planarians as possible and sequence their genomes. So keep your eyes on his lab if you want to do the genomic studies.
I had like another really naive and possibly yeah. So I guess like in mammalian tissues, like kind of like they have the separation between like physiology and cancer pathology, being that like okay, like in physiology we have like cells that are for the most part, I mean some tissues have high degrees of turnovers, but a lot of tissues have a lot of quiescence, right? And sort of like very strict like anatomical conformation that sort of like imposed like structure on the tissue, right?
Yeah.
And like I'm wondering like is a planaria like I mean not yeah, like is it like is it kind of like an odd creature in the sense that almost the whole tissue is actually not like our tissue, but it's already a cancer-like tissue as a whole. And it's just has evolved to maintain itself almost as like a highly plastic sort of, yeah, almost like a cancer-like state that that it's in a different space. I don't know. I mean, that's, and so that almost that it's not that, like, that it's odd that it doesn't have cancer, but that it's all actually like, could you try and push it and like become less regenerative? And would that teach us something about, how is cancer working? I don't know. Like, it's a very odd
Yes.
question, and you
Yeah,
can
yeah.
say like,
So
oh, I don't
I
know.
kind of like this idea. So I actually think that regeneration is a cancer-like state,
Mhm.
especially in planarians. I mean, if you look at the gene expression profile of the cells in a blastema or during like the first 4 days after an amputation, those cells upregulate everything that I would expect cancer cells to upregulate. You can see EMT, you can see oncogene expression, you can see just elevated levels of DNA damage, error-prone DNA damage repair, like anything you associate with a cancer cell, you
Mhm.
see upregulated during regeneration. However, the animal actually can't regenerate all the time. So like homeostasis, I actually think all of those signals are dampened and even inhibited. And there are active tumor suppression pathways in planarians. So this is sort of other half of the lab's work. But essentially what we found is that if you even put a little bit of extra DNA damage into planarians, they don't regenerate anymore. And they actually turn on a bunch of innate immune signaling pathways and tumor suppression mechanisms to prevent the stem cells from migrating, differentiating, and building missing tissues. And so I think an organism that was in a constant state of cancer wouldn't still have all of those mechanisms. So yes, I agree. It's an invertebrate. It's loosey-goosey. Like the inside of the worm does not have sort of a stereotyped architecture. It just has a lot of sort of soft mesenchymal tissues that sort of grossly pattern the organ systems. But I do actually think there is a difference between a stem cell at homeostasis, just maintaining a planarian, and a stem cell that has been given permission to regenerate missing tissues.
Mhm.
And so I think that's actually, I mean, it kind of makes sense. You wouldn't want an organism to just constantly be building random tissues all the time. And so I think understanding this switch will help us understand how planarians turn on a little bit more plasticity specifically during regeneration and then turn it back off again and help everything settle back into sort of a more homeostatic state. But the fact that they can switch so easily is definitely a unique aspect of planarians. Like it's very easy for them to switch into a sort of temporary cancer-like state and then switch back.
Mhm. No, I mean, it's super cool. Like, it's not like
Yeah. But
yeah,
I think,
it's
I mean, I think there's lots of reasons you wouldn't want that in a
all the
mammal,
time.
right?
Yeah, 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 yeah.
Like, I mean, and we have other adaptations to like ensure our progeny do good things, and we live significantly longer, and like we have all these complex memories and social structures that, if our brain could just get like lopped off and regrown all the time, like our social structures wouldn't work, right? Like our
Yeah.
all of our beautiful evolutionary adaptations to learn and build tools and like collectively come together as a society, like I don't think that would be resilient to like this level of remodeling that's always happening in planarians.
Mhm.
And so I think there's advantages to being more stable because it allows you to build up these like complex behaviors and systems as an organism. Um, and I think there's a limitation to what planarians can do, right? Like they can't necessarily develop those. So I think we want to maintain all of our strengths as a model system slash species. Um, but
Mhm.
maybe we can learn a little bit about how to sort of transiently activate growth, rejuvenation, plasticity in a safe way from planarians and then try and translate that to mammals. Or at least that's how I'm thinking about it.
Yeah, no, it's very, very cool.
Yeah.
Yeah, I guess I find it interesting also, like you're mentioning this switch between regeneration and homeostasis, but it seems like even in homeostasis, like when the animals are very well fed, they're going to continue growing to maybe what, like 10 times the size of when they're more starved. So is there a switch there also between, or like nutritional control, I guess, of
Yeah. Yeah. So there's
the growth of normal tissues?
Yeah, so there's definitely nutrient-based coupling between sort of nutrient availability and most likely insulin signaling, sort of canonical nutrient signaling, but it's probably insulin signaling and mTOR if I had to throw it out there. And all of those signaling pathways are conserved. And so when you feed a worm, there's a burst of proliferation. And we think that that's mediated by intestinal signaling, absorption of nutrients into the intestinal tissues, secretion of those nutrients into the surrounding stem cell compartment. So you get this burst of mitotic events that sort of lasts, I mean, it peaks around 2 to 3 days, like 1 to 3 days after you feed, and then it sort of declines over the next week or two. And interestingly, if you irradiate a worm after feeding, there's so many more stem cells that actually they it can be basically the number of stem cells that survive the same dose of ionizing radiation is significantly higher. And so, you can get a lot of variability in stem cell behaviors during like in a starved animal versus a well-fed animal. And then whenever you feed the worm, yeah, that burst of proliferation eventually leads to an increase in tissue, growth of the body plan, and sort of the appropriate proportional addition of cells to all of the right places in the animal. And not every tissue scales the same way. There's actually, I think Daniel Lobo's lab has been doing some like mathematical modeling of different tissues, but like the brain and the intestine don't scale similarly. We're interested in doing some like analysis of how, because there are these size-dependent changes in the worm that correlate with asexual reproduction, it would make sense that underlying tissue heterogeneity, composition, signaling is contributing to that. And so we think, you know, not everything gets added in the same proportion. I feel like basically there's priorities, like they build this minimal set of tissues, and then as the worm grows, they kind of expand some things, they elaborate on the brain structures and the capacities of the brain. Um, and they maybe like the patterning of some of the mesenchymal space also changes in a small worm versus a large worm. And so, the big worms eat a lot compared to small worms. Like just like and like they get thicker in addition to getting longer. So the mass size change is actually pretty huge between a 20 millimeter worm and a, you know, 3 millimeter worm. And in the sexual animals, the germline is developed at a specific size and nutrient state. And so in the sexual animals and in other planarian species, there are size-dependent organogenesis signaling mechanisms as well. So there's definitely like a sort of inherent, there's a lot of growth-dependent things that can happen, which I guess is actually, I mean, it's the same for us, right? Like when we reach a certain size/age, we develop organ systems as well. So
Blair,
makes,
I know we booked you tier 11, but if you have a few more minutes to spare, can I ask more
yeah,
questions?
yeah, sure.
Okay. So I'm just thinking about a parallel between the asexual production and the amputation response, like amputation-induced regeneration response. Obviously, I can see the downstream process. Maybe there are a lot of shared mechanism to regenerate the tissue, right? But the asexual production is almost like a planned process, and they're actively doing so, whereas the amputation is passively doing so. They have to just put things together in response to this emergency of amputation. And I just wonder if what is going on in this planned process of sexual production in order to plan for the fission, like playing like when they fall off and to regenerate. Do they before that is any molecular? I don't know anything that is happening, molecular level or structural level or even epigenetic level for the stem cell nearby do they start to prepare them, prime them to undergo this active process of
Yeah.
fission?
The fundamentally we don't know. Like it's a very good question. I think that through fissioning, you're going to definitely get a head start on some of the earliest stages of regeneration. So most of the internal tissue gets sort of fragmented and segmented and like mechanically squished apart. And so there's going to be a bunch of wound-induced signals and injury-induced signals that happen. Additionally, the body wall musculature rips before the epidermis ever pinches off the wound. And we know that there are wound-induced injury, like injury-induced genes in the muscle specifically. So it would not surprise me if progeny produced through asexual reproduction had anywhere from like a 1 to 6 hour head start on a lot of the signaling pathways. Also, the wound produced by asexual reproduction is really small. So there's like this tiny little purse string that gets pinched off, and like the epidermis just breaks a little bit. And so you can imagine that like the risk of infection or tissue loss is actually significantly less with an asexual reproduction event than with an amputation, where you have this like big gaping wound that you have to like try and close using the epidermis and the muscle itself. And so the risk of some sort of pathogen getting in during that wound healing process is much higher. So there are definitely advantages to asexual reproduction. In terms of like the mechanisms downstream of that, we have not looked. I mean, luckily, I mean, we now have been playing around with regulation of asexual reproduction, and we have found ways to like inhibit asexual reproduction in animals without a genetic manipulation. And then we can release them from that, and they all want to fission really badly all at once, which is nice. So you could actually do a systemic comparison of, we're going to collect fission fragments at all of these early time points after the fission fragment is produced. And we're going to profile the stem cells. We're going to profile cell-cell signaling mechanisms at these early time points with amputation versus asexual reproduction. You could do the comparison. I think we now have abilities to do that, but we haven't done it yet.
Do the fission from the asexual production regenerate faster than the amputated fragment of similar size or like similar position?
I don't think so, but I haven't actually looked. You would maybe anticipate that like wound closure would be faster, for sure. And then you might get like slightly earlier wound-induced gene expression. And then the real question is like in terms of metric, like when does the animal start eating again? Or when do you first see a neuron, like a photoreceptor cell? You really have to go in and kind of carefully time it to know. Like in terms of like days, I don't think so. I think it would probably be hours. Yeah.
Like how I just wonder if the sexual production is primed for them to quickly regain the body plan, like the structural intestine, all this thing, reorganization, whether that process happen faster than just you randomly amputated.
As far as I can tell, I haven't seen large differences, like multiple days faster. It wouldn't surprise me if it was a little bit faster. But also there's even just like where you take a fragment from can affect how quickly it builds missing tissue. So, if you cut the worm close to the head, it's actually going to be able to rebuild the head in like 6 to 7 days. But if you cut a worm really close to the tail and very far away from the head, it might take it 10 days to build head tissue. And it's just because it has to do so much remodeling, and there are fewer stem cells that are already primed to build that tissue. And so you kind of have to rebuild the right progenitor pool first and then rebuild the tissue that's missing. And so, yeah, fission progeny, depending on where they are in the body plan, will also build head structures or pharynx structures at different rates.
You noted in your summary slide, the question here, light exposure also inhibit the sexual reproduction, but we all see your videotaping them and there's light there. So what kind of light is that?
Yeah, so we used very dim light to get them to asexually reproduce. And the really pretty two-sided movie it actually took them 3 days to fission in that environment. But if I put a worm like in an incubator on its own, that same worm probably would have fissioned 3 times in the same period. So there's almost like an acclimation time where they like kind of they want to fission, but they don't want to fission in light. But eventually they give up and are like, fine, I'm just in a light environment. I guess I can't get away from it. So I guess I better get on with it.
So it has to act through the eye or the pigment, like the all the visual neurons, or it's just directly sensing light for the
So
pig.
there are the photoreceptors in the brain actually form these like pigment cups that sense directionally where light is coming from, and the worm will swim away from light if it can. And if it's in a really well lit environment, it will actually like try to invert its body and swim along the surface tension on the top of a dish just because it doesn't want its photoreceptors to get hit by light. And so those main eye spots, like pigment cups, really drive a lot of the motility of the animal and sort of light avoidance. However, you can cut off the brain, and there are rhodopsin molecules that are patterned along the sort of peripheral nervous system as well. So even a regenerating tail fragment will very, very slowly creep away from light. It's just not nearly as coordinated as a full worm.
Cool. I wonder if anyone else have any further question. It's so lovely to have you here. So Anita, you want to say something?
Oh no, I just wanted to thank you for your time. No, no, sorry.
It's
I really enjoyed it. I thought you gave a really beautiful seminar as well.
it's
So
fantastic.
both things were really wonderful. Thank you so much.
Great. I'm glad I could come.
Yeah, it's very illuminating. I really enjoyed your talk, and I
Yeah.
really learned a lot from you. I'm very excited about what is going on. I mean, maybe before we end, like since we are recording and sharing with students, I wonder if you want to share what you feel excited about the environment and Baylor, how it maybe you want to seek some opportunity of the environment, new environment to do something new, something you were not doing before.
Yeah. One of the reasons I came to Baylor was that they sort of saw the potential of planarians as a discovery system for anti-cancer and anti-aging mechanisms. Most of my colleagues work in mammalian systems, and disease models. And, there are definitely some strong and proud invertebrate people here, but a lot of zebrafish and mouse and human studies. And we have a really close collaboration with several hospitals here in the Texas Medical Center. So yeah, I mean, our core facilities are amazing. I only wanted to go to a place that had similar resources as the Stowers Institute. And so I think, moving forward, we're doing a lot of basic characterization, discovery of new mechanisms and planarians, but realistically, the second half of every aim is basically to test if those mechanisms are still at play in mammals, in either an organoid system or a mouse model, or maybe even a human organoid model. And so, I'm in the right place with the right people and the right collaborators to do that work. And so I think, that is the next step. And so if there are folks that are like a little nervous about doing a postdoc in a planarian system, but would be excited to test if planarian mechanisms could help rejuvenate or increase the resilience of mammalian stem cells, then that's sort of, I think, the next horizon for projects in the lab.
Okay.
Thank you very much. Yeah,
Yeah.
really enjoyed talking
Thank
about
you
this
so
subject.
much.
Yeah.
Bye
All
bye.
right.
Bye
Thanks.
bye.
Bye.
Bye.